Greetings everyone. Today we're diving into single-cell RNA sequencing analysis in OR. You might be wondering, what are the differences between single-cell RNA sequencing and bulk RNA sequencing? With bulk RNA sequencing, if you measure gene expression from group of cells, you get just one average value per gene for all of those cells, missing out on individual differences. In contrast, single-cell RNA sequencing measures gene expression for each cell separately, revealing how each one varies, and allowing us to study the diversity within the population. Without further ado, let's get started.
Firstly, we upload our libraries. We have dplyr, Seurat, and Patchwork. We use dplyr for data manipulation tasks like filtering, selecting, and summarizing data. And we use Seurat for performing the single-cell RNA sequencing analysis workflow. And we use Patchwork for combining multiple ggplot2 based plots into a single plot. In single-cell RNA sequencing analysis, we frequently generate visualizations like phylum plots and scatter plot. Now let's load our data and create our ScRNA-seq. The data is PBMC, which stands for Peripheral Blood Mononuclear Cells. This type of data typically refers to a collection of blood cells that are extracted from the peripheral blood of a person. So here we created a variable of the data called the PBMC.data. And then we used read10x, which is a function that understands the specific format of 10x genomics data. And inside, we wrote data.dir. Dir comes from directory, which specifies where our data files are located on the computer. Then we initialized our ScRNA-seq. We wrote PBMC as a variable, and we used the function CreateSeuratObject. CreateSeuratObject takes your raw data and set it up for further analysis, and then counts. We provide the raw count matrix that we have just loaded, which is equal PBMC.data, and then project. Here we call it PBMC 3K. Minimum cells is 3, and minimum features 200. Minimum cells, this means only include genes that are found in at least this many cells. So setting it to 3 means we only keep genes found in 3 or more cells. This helps get rid of genes that aren't useful. And for minimum features equal to 200, this means we only keep cells with 200 or more detected genes. This helps remove cells that don't have enough data to be useful. So let's run our code. So as you can see, we got object of class Seurat. This indicates that we have created an object from the Seurat class, and then 13,714 features across 2,700 samples within one assay. The Seurat object contains 13,714 features, which is genes, and 2,700 samples, which is cells, across one assay, and then active assay RNA. Now what does our data in count matrix look like? We wrote PBMC.data and we put 3 genes that we are interested in among 30 cells. Let's run our code. We have 3 times 30. This means that the matrix has 3 rows and 30 columns, each row corresponding to one of the genes. For example, here CD3D gene, TCL1A, MS4A1, and then 30 columns. We can also see sparse matrix of class DGC matrix. This indicates that the matrix is stored in a sparse format. Sparse matrices are used to efficiently handle data that contains many zeros, because as you see, all these dots are representing zeros. And since most values in the single-cell RNA-seq matrix are zeros, Seurat uses a sparse matrix representation whenever possible. This results in significant memory and speed savings. We have 2 types of matrices, dense matrix and sparse matrix. Dense matrix is a matrix that stores every value, including zeros. It is useful for operations that need all values present but uses more memory. But regarding a sparse matrix, it's a matrix that only stores non-zero values and their positions. It saves memory and is efficient for large datasets. So now let's calculate the size of our data when converted to a dense matrix and sparse format. So for example here, we created a variable called dense size, and then object.size, and then we used as.matrix, and then we put our data. The purpose of using as.matrix is that it converts our sparse matrix into a dense matrix. Let's run our code. As you can see, we have about 709,591,472 bytes. Let's try to run our sparse matrix. As you can see, it's about only 299,005,192 bytes. And if we make a simple calculation, dense size over the sparse size, we get a value of 23.7 bytes. This indicates that the dense matrix uses approximately 23.7 times more memory than the sparse matrix. So in single-cell RNA sequencing data, it often has a lot of zeros. Converting to a dense matrix can help you understand how much memory would be required if all zeros were stored explicitly. And working with dense matrices can be slow and memory intensive for large datasets with many zeros. Now let's go for the QC quality control and cell selection step. Here we will evaluate several key matrices to ensure data quality. First, we will check the number of unique genes detected in each cell, because cells with very few genes might be low quality or empty droplets, while those with unusually high gene counts could be doublets. Next, we will look at the total number of molecules detected in each cell, which correlates with the number of unique genes and help identify cells with abnormal counts. Then we will calculate the percentage of reads mapping to mitochondrial genes using the PercentageFeatureSet. High mitochondrial content can indicate dying or low quality cells. These quality control metrics help us filter out problematic cells and ensure that only high quality cells are used for further analysis. So we write the name of our data, PBMC, and then we put the %.mt or mitochondrial. The syntax is used to add or modify metadata in the Seurat object. %.mt is a new metadata column being added to the PBMC object. And then we use this function, which is PercentageFeatureSet. And inside the function, we put our data, PBMC, and we write pattern, and then empty. The PercentageFeatureSet function, it calculates the percentage of counts originating from a specific set of features. In this case, mitochondrial genes. But why this step is useful? By adding the percentage of mitochondrial gene counts to the metadata, we keep track of the mitochondrial content for each cell. In addition, data filtering. This information helps in identifying and filtering out low-quality cells that may have mitochondrial contamination. So let's run our code. In Seurat, QC metrics are stored in the object's metadata. When we create a Seurat object with CreateSeuratObject, it automatically calculates and stores metrics like the number of unique genes and total molecule counts in this metadata. To view these metrics, we can access the metadata slot of the Seurat object. For example, we can write head and then PBMC at meta.data and we put 5 to extract the first 5 cells. Let's run our code. As you can see, we got our cells, the 5 cells, and our label or identification or ID, then the in-count RNA, which is the total RNA molecules detected in each cell. For example, in this cell, we have 2,419 RNA molecules, and for this sample, we have 4,903 molecules, and so on. In addition, we have the in-feature RNA. In-feature is the number of unique genes detected. So for example, first cell, we have 779 genes. Second cell, we have 1,352 genes, and so on. And the last column, which is the column that we have added, which is %.empty. Here, the percentage of RNA reads mapping to mitochondrial genes. So for example here, 3.01, this means 3% for the first cell, and 3.7 for the second cell, and so on. So now let's visualize the QC metrics using violin plots. We write VLN plot, and we put our data, PBMC, and features. We choose feature RNA, in count RNA, and %empty. The number of columns is 3. Now let's run our code. And here we have it. We got our plots. We have the in-feature RNA plot, in count RNA, and %.empty, or mitochondrial. For example here, in-feature RNA displays the distribution of unique gene counts across all cells. For example here, the y-axis represents the number of unique genes detected. So for example here, 1,000, 2,000, and 3,000. And for the x-axis, it shows the dataset name, which is PBMC3K. Each black dot on the plot represents a single cell's unique gene count. Most of the black dots are clustered under 1,000 on the y-axis, indicating that the majority of cells have fewer than 1,000 unique genes. And regarding the second plot, which is in count RNA, it shows the distribution of total RNA molecules detected across all cells. And the y-axis represents the total number of RNA molecules within values 5,000, 10,000, and 15,000. Each black dot represents a single cell's total RNA count. Most of the black dots are concentrated around 2,000 to 3,000 or 2,500 on the y-axis, indicating that the majority of cells have total RNA counts within this range. From this plot, we can understand the distribution of RNA counts across cells and can highlight any potential inconsistencies or issues in our data. And now, the third plot, which is %empty. As you can see, it illustrates the distribution of mitochondrial RNA percentages across all cells. The y-axis here represents the percentage of RNA reads mapping to mitochondrial genes, with values 5%, 10%, 15%, and 20%. Each black dot on the plot corresponds to the mitochondrial RNA percentage for a single cell. Most of the black dots are clustered around 2 to 3%, indicating that the majority of cells have mitochondrial RNA percentages in this range. This plot helps us assess the level of mitochondrial contamination in our cells and identify any potential quality issues. Now let's visualize the relationships between different quality matrices in our data to better understand how they correlate with each other. So we create a variable called plot1. We use feature scatter to make a scatter plot. We put our data, PBMC. Feature 1 is the in count RNA. Feature 2 is the %empty or mitochondrial. The second plot the same, but instead of %empty, we put in feature RNA. And here together the two plots. The feature scatter is function to create scatter plots that show relationship between two features or metrics in our data. So let's run our code. As you can see, we got our plot. The first plot is %empty versus in count RNA. This plot shows the relationship between the total number of RNA molecule in count versus the %empty or mitochondrial, which is the percentage of mitochondrial reads It helps us to see if there's a correlation between the amount of RNA and mitochondrial contamination. For example, high mitochondrial percentages might be associated with low read counts. As you can see here, we have the highest percentage, about 10%, or from 8 to 10%. And then when the in count increases, we have our curve here, so the percent empty decreases. As we can see above the plot, we have a value of -13. This indicates a weak negative correlation between the two features being compared, which are in count RNA and percent empty. In simpler terms, it means that there is a very slight tendency for the percentage of mitochondrial RNA to decrease as the total RNA count increases. However, this relationship is not very strong, and there are likely other factors influencing the expression of these features. And regarding the second plot, it presents the relationship between the in-count RNA and in-feature RNA. For the y-axis, or the in-feature RNA axis, we have 1000, 2000, and 3000 values. And for in-count, we have the same 5000, 10,000, and 15,000. As we can see, the higher the in count, the higher the in feature. And here, for example, the highest dot, it exceeds 30,000 on the y-axis, or the number of unique genes, and about 15,000 on the x-axis, which is the total RNA molecules. This means that the cell has an unusually high number of unique genes and a total RNA count. This indicates that it likely represents an outlier or doublets, and should be investigated further. The value or the correlation coefficient above the plot is 0.95. This indicates a strong positive correlation between the two features being compared, which are the in-feature RNA and the in-count RNA. In simpler terms, it means that there is a very clear tendency for the number of genes detected in the in-feature RNA to increase as the total number of RNA transcripts in count RNA increases. This suggests a strong relationship between these two features. Now let's visualize QC metrics and make some filters. For example, we filter cells that have unique feature counts over 2500 or less than 200. Also, we filter cells that have more than 5% mitochondrial counts. So, we write PBMC and we use the subset function. We put PBMC, and for subset, we put in feature RNA, we make it more than 200, and in feature RNA, we put it less than 2500, and the percent empty, we make it 5%. Let's run our code. By using subset function, we created a new Seurat object from the existing PBMC object, based on the specified conditions that we have entered. Let's check our output. We have an object of class Seurat, 13,714 features across 2,638 samples, within one assay. As we agreed before, the features represent the number of unique genes, and for samples, it represents the number of cells. And there is assay 1, which is a set of data, in this case RNA counts. Now let's normalize our data. After removing unwanted cells from the dataset, the next step is normalization. We use the default global scaling method called log normalize. This method normalizes each cell's gene expression by dividing the total expression for that cell, multiplying by a scale factor, which is the default is 10,000, and then applying a log transformation. Let's run our code. We can also use this command to normalize our data, which is NormalizeData, and we put our data, PBMC. After normalization, the next step is identification of highly variable features. We calculate which features show significant variation between cells that are highly expressed in some cells and low expressed in others. So we make the PBMC variable and we write find variable features function. Inside we put our data and then selection method VST, and we put number of features 2000. The selection method equal VST. This uses the variance stabilizing transformation method to select highly variable features to identify genes that exhibit significant variability across different cells. These genes are often of interest in single-cell RNA sequencing because they can reveal important biological differences or signals. But how it works? This method normalizes the variance of gene expression levels across samples or cells. This helps to stabilize the variance so that it's less influenced by the mean expression level, and then ranks genes based on their variance, allowing us to select those with the highest variability. And then we identify the 10 most highly variable genes. We use variable features and we put our data and we write 10. And then we plotting the variable features by writing plot 1, variable feature plot, and we put our data for plot 1. And for plot 2, label points. Label points add labels to the plot created by variable feature plot. Plot equal plot 1 to specify that we are adding labels to plot 1. Points equal top 10 specify that labels should be added to the top 10 most variable genes. And then ripple equal true ensure that the labels are adjusted to avoid overlapping and to make the plot more readable. And finally, we combine plot 1 plus plot 2. Now let's run our code. And here we have it. We have plot 1 and plot 2, standardized variance versus average expression. Here, as you see, we have the highest or the most variable genes. As we can see, the highest variable gene is PPBP, and then LYZ, and so on. For the non-variable count, 11,714, and for variable count, 2,000. So these labeled genes exhibit significant variability in expression levels across different cells in our data. This means that they are expressed at different levels in various cells, making them stand out in terms of their variability. Focusing on these genes can help reveal important biological differences or patterns. Now let's go for the next step, which is scaling our data. This step is crucial pre-processing step before applying dimensional reduction techniques like PCA. Firstly, we make a variable called all.genes and put row names of the PBMC. This retrieves the names of all the rows in the PBMC object. In this case, it's the name of the genes. And then we perform the ScaleData function, which is used to scale and center the data for each feature or gene. And then we put features equal all.genes. Now let's run our code. By setting features equal all.genes, we are telling the function to apply the scaling to all genes in the dataset, not just the highly variable ones. And here, for example, we could regress heterogeneity associated with cell cycle stage or mitochondrial contamination. We also use ScaleData function and we put our data and then we write vars dot to regress and we put %empty. And now let's perform our linear dimensional reduction or PCA. Linear dimensional reduction is a technique used to make complex data easier to handle and understand. In single-cell RNA sequencing, we often deal with data from thousands of genes, which can be overwhelming and difficult to analyze directly. To simplify this, we use linear dimensional reduction methods like principal component analysis or PCA. The goal is to reduce the number of dimensions or features while still keeping the most important information. The benefits of this step are significant. By reducing the complexity, we can more easily visualize the data and identify patterns. It also speeds up the analysis because working with few dimensions is less computationally intensive. Before performing dimensional reduction, it's very important to scale the data. Scaling ensures that all features or genes in the case of single-cell RNA sequencing contribute equally to the analysis by adjusting them to have a mean of 0 and a variance of 1. This step is crucial because without scaling, features with larger values or greater variances could dominate the results, skewing the analysis and make it difficult to identify true patterns. Now, why we call it linear? We call it linear because they simplify complex data by using linear combinations of the original features. For example, in this context, a linear combination means combining features in a way that adds them together with some weights. For instance, if we have two features, A and B, a linear combination could be 0.5 times A plus 0.3 times B. The coefficients 0.5 and 0.3 are weights that determine how much each feature contributes to the new dimension. So, we write PBMC variable and then run PCA to make the principal component analysis plot. We put our data, PBMC, features equal variable features, and we put object PBMC. The variable features object equal PBMC. This part retrieves the most variable genes identified in the data or the PBMC Seurat object. These are the genes that show significant variation across cells and are thus considered more informative for the PCA. Now let's run our code. As we can see, we have PC1 positive, negative, PC2 positive, negative, PC3, PC4, PC5, so we have 5 PCs. Each PC has the positives or the genes which indicate positive loadings and some genes negative or negative loadings, and the same for every PC. For example, PC1, it captures the most variation in the data. It represents the direction in which the data varies the most. And PC2, it captures the second most variation, and so on. So PC1 might show that some cells have high levels of certain genes, while others do not, revealing a measure pattern. For PC1 positive features, these are genes that contribute strongly to the positive direction of PC1. They show higher expression levels in cells that score high on PC1. And for negative features, these genes contribute to the negative direction of PC1. They show higher expression levels in cells that score low on PC1, and so on. Genes with positive loadings on principal component tend to be co-expressed, meaning they are often found at high levels in the same cells. This suggests that these genes may be involved in similar biological processes or regulated by common pathways. Conversely, genes with negative loadings on a PC are often inversely related. Cells that have high expression of one of these genes typically show low expression of the others, suggesting that these genes might be involved in opposing or antagonistic processes. So now let's examine and visualize our PC. We write print PBMC and we put PCA dims 1 to 5 in features equal 5. Let's run our code. As you see, we put in feature equal 5, so we got 5 genes per each. And dims the dimension or the number of the PCs, 1 to 5. So got 5 PCs. And now let's go for the loading plot. It has the top genes associated with each principal component. Which genes are important for PC1? Also, we can see the relation between the genes and the principal component, as here is positive related as direct or negative as indirect. So we write VIZ dim loadings and we put PBMC dimensions 2 and reduction equal PCA. Let's run our code. As you can see, we put dimension equal 2, so we got it for PC1 and PC2. The top genes are associated with each principal component. We can see also which genes are important for PC1. Also, we can see the relation between the genes and the principal component, as here is positive related as direct, or negative as indirect. So, for example, CT3 here. The high positive value for CT3 implies that cells with high expression of this gene tend to cluster together on positive side of the plot. This suggests that CST3 might be differentially expressed in a specific cell type, developmental stage, or under particular conditions. And here, as we can see, these genes are contributed to the negative side with the PC2, and these genes to the positive of the PC2. Now let's plot scatter plot of the cells using PCA. So we use dim plot. This will show how the cells are distributed based on the principal components. To keep the plot clean, we will remove the legend with no legend. Let's run our dim plot. As you can see, it's a scatter plot of the cells using PCA. This shows how the cells are distributed based on the principal components. As you can see, we have two big groups, here and here. Group 1 located on -5 on PC1 here and about 0 on PC2. This cluster represents the cells that have lower values on PC1. These cells share common gene expression features that leads them to be grouped together on this side of the plot. And for here, this cluster positioned around 12 on PC1 and the same 0 or like 1 or 2 for PC2. This cluster represents cells with higher values on PC1. These cells have a different set of gene expression features compared to those in group 1, leading them to be distinctly separated in this part of the plot. The separation into two clusters suggests that there are distinct biological differences between these groups of cells. These differences could be due to various factors like cell types, states, or conditions. And we can also see that here we have a small cluster, with PC1 around -2 and PC2 about 10 or 9, and so on. So now let's use dim heat map to explore the primary sources of variation in our data. So we write dim heat map, and we put PBMC, dims 1, cells 500, balanced equal true. We put dim 1 to specify that heat map will focus on the first principal component or first PC or PC1. Cells equal 500 limits the heat map to display the top 500 cells with the most extreme PCA scores, both high and low, for faster plotting. And finally, balanced equal true ensures that an equal number of cells with positive and negative PCA scores are displayed, giving more balanced view of variation on both ends. Let's run our code. As you can see, we got our heat map. We have genes are ordered according to their PCA scores. It shows the expression levels of the most influential genes contributing to PC1. Each row represents a gene, and the heat map color intensity indicates the level of expression. The genes are ordered by how much they contribute to the variation in PC1, which helps in identifying which genes play a significant role in separating different cell populations. In particular, dim heat maps allows for easy exploration of the primary sources of heterogeneity in our data, and can be useful when trying to decide which PC to include for further downstream analysis. We can also make heat maps for 15 PCAs. And instead of put dim 1, we put 1 to 15. Let's run our code. As we see now, we have one plot containing 15 heat maps, each representing a different principal component. Someone might ask a question: which PCs should we include for further analysis? Should be 1, 2, or 15? Here the elbow plot comes into play. An elbow plot helps us determine how much variance each PC explains. By examining this plot, we can identify a point where the explained variance starts to level off the elbow, which gives us a clue about the optimal number of PCs to use. So let's make our elbow plot. We write elbow plot and we put our data. Let's run our code. And here we have it. As you can see, here is the number of PCs and here is the standard deviation. Based on the elbow plot, we will choose 10 because the plot shows a noticeable elbow or inflection point around the 10th PC. This is where the amount of explained variance begins to level off, meaning that after PC 10, here, the remaining components contribute much less to the overall variance in all of the data. Now let's cluster the cells to identify distinct groups based on their gene expression profiles. First, we will build a k-nearest neighbor using findNeighbors function. So, we write PBMC, findNeighbor, we put PBMC, dims, dimensions from 1 to 10, and then PBMC, find the clusters, PBMC resolution, equal half. This algorithm constructs a graph where each cell is connected to its closest neighbors based on the selected principal components. Then the algorithm calculates the shared nearest neighbor or SNN graph, which refines the k-nearest neighbor graph by adjusting edge weights based on the overlap of local neighborhoods. Shared nearest neighbor graph is to make the connections more accurate. The algorithms look at how many neighbors each cell shares with others. If two cells share a lot of the same neighbors, they connected more strongly. And for the overlap of local neighborhoods, this means adjusting the connection between cells to reflect how similar groups of their neighborhoods are. So let's run our code. So as you can see, computing nearest neighbor and computing SNN. Number of nodes is 2638 and number of edges 95,893. The number of nodes here indicates the total number of cells included in the analysis, and for the number of edges shows the total number of connections or edges between cells. And then running Louvain algorithm. This algorithm iteratively groups cells into clusters to optimize modularity score. And the maximum modularity in 10 random starts equal to 0.87. The best modularity score achieved during the clustering processes. Higher values indicates better defined clusters. And then the number of communities 9. This is the final result is 9, which indicates 9 clusters or 9 groups of cells. Now let's look at the cluster IDs of the first 5 cells. So we write head, ident, PBMC of 5. Let's run our code. As you can see, we have 5 cells, 1, 2, 3, 4, 5, and we have numbers 0, 3, 2, 1, 6. The output lists the possible cluster IDs, which ranges from 0 to 8. This means that we have 9 clusters in total, same as we said. Now let's run nonlinear dimensional reduction using UMAP. UMAP and Disney help in visualizing the data by placing similar cells closer together in the plot. This should make cells that belong to the same clusters, as identified by our previous clustering step, appear together. It's important to remember that while these visualizations are useful for exploring data, they have limitations. They focus on preserving local structures, but might not accurately reflect the global relationships within the data. Therefore, while UMAP and similar techniques are valuable for visual exploration, it's crucial to combine them with other analysis for complete understanding. So, PBMC, we run UMAP, PBMC dimension, we put 10. So, let's run our code. Now, let's visualize our plot by writing dim plot, PBMC, reduction equal UMAP. Let's run our code. And here we have it. As you can see, the plot displays cells positioned in a 2D dimensional space based on UMAP reduction. Each dot represents a cell, and the positioning of the dots reflecting the similarity with the gene expression profiles. So, for example, we have here a cluster, and we have here a cluster, and here cluster, and so on. Cells are color-coded according to their cluster IDs. As we can see, we have 0, 1 until 8, because we have 9 clusters. Each color corresponds to a specific cluster, and the clusters are grouped closely together based on the similarity of gene expression profiles. This visualization allows us to see how the cells are organized into these clusters, and it helps in understanding the overall structure and relationships in the data. Now we should identify cluster-specific biomarkers and determine which genes are uniquely expressed in each cluster. In this case, we have to use findAllMarkers function to automatically find these genes across all clusters. So, we write clusters2markers to find all markers of cluster 2, for example, and find markers, put PBMC, ident, we put 2 for cluster 2, and hit cluster 2 and n equal 5. Let's run our code. And here we have it. As you can see, we have our genes here, the 5 genes, and we have p-value, average log to fold change, PC1, PC2, p-value adjust or adjusted p-value. Regarding the p-value for the differential expression of each gene, a very small p-value close to 0 suggests that the gene is likely differentially expressed between the specified cluster and the other cells. And for the average log to fold change, it is the average log to fold change of gene expression between the clusters of interest and the other cells. Positive values indicates higher expression in the cluster compared to the other cell. For example, here, the first gene, which is IL32, is 1.2, meaning it is expressed more in the cluster compared to the others. And then PC1, the percentage of cells within the cluster that expresses the gene. For the first gene, IL32, PC1, it's about 0.95, meaning 95.1% of cells in the cluster express IL32. And then PC2, the percentage of cells outside the cluster that express the gene. For the first gene, it's 0.47, meaning 47.5% of cells outside the cluster express this gene, or IL32, and so on. And finally, the p-adjusted value, accounting for multiple comparisons. This value is corrected to reduce the risk of false positives. For the first gene, IL32, the adjusted p-value is extremely small, indicating very strong evidence that IL32 is differentially expressed. And now, let's find the all markers distinguishing cluster 5 from cluster 0 and 3. So, cluster 5 markers, we write find markers, and then PBMC, ident 1 equal 5, ident 2, 0 to 3, and then hit clusters 5, n equal 5. The find markers function, as we said before, it is used to identify genes that are differentially expressed between two groups of cells. In this case, it's comparing cells from cluster 5 against cells from cluster 0 and 3. And for ident 1 equal 5, this specifies that cluster 5 is a group of interest. We want to find genes that are specifically expressed in cluster 5. And for ident 2, C0 and 3, this specifies that the comparison group includes cells from clusters 0 and 3. Essentially, the function will compare the expression levels in cluster 5 with the combined expression levels from cluster 0 and 3. Let's run our code. And here we have it, 5 genes, MB value, average log to fold change, PC1, PC2, and P adjusted value. What if we want to find the markers for every cluster compared to all remaining cells? Report only the positive ones. In this case, PBMC markers, find all markers, we put PBMC, only positive or pose, equal true, PBMC markers, and then group by cluster, and then the player, filter, average log to fold change, more than 1. Let's run our code. We have our genes, P adjusted value, PC2, PC1, average log to fold change, and P value. Now we should refine our analysis to identify specific markers that distinguishes cluster 0 from other clusters. To enhance our understanding, we use Seurat's various tests for differential expression, which can be set using the test to use parameter. For instance, the ROC or Receiver Operating Characteristic test evaluates the classification power of each marker, providing a score that ranges from 0, random, to 1, which is perfect. So to implement this, we need to use find markers. So we make cluster 0 markers, find markers, and then PBMC ident1 equals 0, log fold change threshold to 0.25, test use ROC or Receiver Operating Characteristic test, and only positives equal true. So ident1 equals 0 specifies that we are focusing on cluster 0, and log to fold change 0.25 sets the minimum fold change of a gene to be considered significant. Genes must have at least 0.25 fold change to be included, and we choose the ROC test to assess the ability of each gene to distinguish cluster 0 from other clusters, and only .pos or positive equal true limits the result to genes that are upregulated in cluster 0. Now let's run our code. Now we need to visualize the expression levels of specific genes across different clusters to better understand how the genes are among the clusters. This helps us identify which genes are uniquely expressed or differentially expressed in particular clusters. To do this, we will use the Vlim plot function, which creates Vlim plots showing the distribution of gene expression levels. So, Vlim plot, PBMC, features, and we put our genes that we are interested in MS4A1, CD79A. Let's run our code. And here we have it. As you can see, we have two plots. The y-axis is the expression level. This axis shows the expression level of the gene. Higher value indicates higher gene expression. And for the x-axis, this lists different clusters from 0 to 8 to make the 9 clusters. And regarding our Vlim plot, it displays the distribution of gene expression levels within a cluster. The width of the Vlim at any given expression level indicates the density of cells expressing the gene at that level. Wider section means more cells have that expression level, while a narrower section means fewer cells. So for example, here in cluster 3, we have the majority. So for example, the plot indicates that in this cluster, which is cluster 3 here, the gene expression is relatively high. The body of the Vlim plot for both genes spans from expression levels between 1 to 4, as you can see from here, 1 to 4. This suggests that within cluster 3, most cells have expression levels of these genes in this range. And the same for this gene, which is CD79E. Within cluster 3, most cells have their high expression levels of this gene. Now we are going to visualize the expression of two specific genes, which are NKG7 and PF4, using row count data from our Seurat object. We will use the blend plot function, but we have to put slot equal counts, and then log equal true. Setting plot equal counts to plot the gene's expression directly from the row counts rather than normalized data, and log equal true will apply a log transformation to the counts. This approach allows us to view the absolute gene expression levels and compare them across clusters, while handling the wide range of expression values more effectively. Let's run our code. And here we have it. We also have here expression level, and here's the clusters, how the majority are here in cluster 4. We have also in cluster 5 and 6 and 7, but the majority are in cluster 4. And for PF4, the majority here in cluster 8. And now feature plot. Feature plot is to visualize the expression of specific genes. Feature plot can display multiple genes at once. This is particularly useful when we want to compare the expression patterns of different genes side by side. By visualizing multiple genes, we can uncover relationships and co-expression patterns that might not be obvious from numerical data alone. By visualizing the expression of marker genes, we can distinguish between different cell types. For example, if we know that gene A is a marker for neurons and gene B is a marker for glial cells, we can use feature plot to see where these genes are expressed and identify the corresponding cell types. So let's run our code. As you can see, the name of our genes, the genes that we have entered, are here: UMAP1, UMAP2, and the expression level. The closer the color to white, the closer the value to 0, and the closer to the blue or dark blue is close to 4. Now let's visualize the top differentially expressed genes for each cluster using a heat map. This step helps us identify which genes are most highly expressed in each cluster and compare their expression patterns across clusters. So, we will write PBMC.markers, group by, cluster, the player, we use filter, average log to fold change more than 1, and we make slice head for 10, which is the top 10 genes, and we call it top 10, and then we put the do heat map. Let's run our code. And here we have it. Up we have the clusters 0, 1, 2, 3, 4, 5, 6, 7, and 8. And here we have the genes. We can also plot the top 3 genes, because as we can see here there are many genes. So instead of n equal 10, we put n equal 3. And we call it top 3. Let's run our code. As you can see now, you can read the genes. Here's the top 3 genes for the all clusters. And here for the all clusters is top 3 genes and top 3 genes and so on. Fortunately, in the case of this dataset, we can use canonical markers to easily match the unbiased clustering to known cell types. For example, we have cluster ID markers and cell type. For cluster 0, we have IL7R and CCR7. And for the cell type, we have naive CD4 plus T. And cluster 1, we have CD14 and LYZ. And for cell type, we have CD14 plus mono, and so on. So for example, in cluster 0, identified by markers IL7R and CCR7. This means that cells in this cluster express high level of IL7R and CCR7, which are characteristic of naive CD4 plus T cells. This means that cells in this cluster express high levels of IL7R and CCR7, which are characteristic of naive CD4 plus T cells. Naive CD4 plus T cells are a type of immune cell that has not yet encountered as an antigen and is in its initial unactivated state. And for cluster 3, it's marked by MS4A1 here, represents B cells. This indicates that cells in this cluster express MS4A1, a marker gene associated with B cells. B cells are a type of white blood cells that is essential for producing antibodies and are involved in the adaptive immune response. So based on the table that associates each cluster with specific cell types, we are going to rename the clusters in our dataset to reflect these cell types more clearly. This renaming process will help us interpret the clusters in a biologically meaningful way. After renaming, we will generate a UMAP plot to visualize the updated clusters. So, we write new cluster ID, C, and we put the name of the cell types, naive CD4T, CD14 plus mono, and so on. And then names, new.cluster.ids, levels, PBMC, and PBMC, rename idents, PBMC new cluster ID, and then dim plot. We put PBMC, reduction, UMAP, and label equal true, and we set size to 0.5, plus not legend. Let's run our code. And here we have it. We got UMAP 1, UMAP 2, and then we have our clusters, the 9 clusters. For example, here we have cluster for NK, a cluster for CD8T, memory CD4T, naive CD4T, MB or B cells, DC, platelet, and so on. For example, here in NK stands for natural killer cells. These are type of lymphocyte, which is a white blood cell that plays a crucial role in immune system. And here, CD4T memory. Generally, CD4T cells, these are a type of T cell that helps regulate and coordinate the immune response. They are often referred to as helper T cells because they assist other immune cells in their function. But regarding the memory CD4T cells, which is here or make this cluster, after an initial infection or vaccination, some CD4T cells become memory cells. These cells persist long-term in the body and remember the pathogen. If the same pathogen is encountered again, memory CD4T cells can respond more quickly and effectively than the first time.
And here we've reached the end of our analysis. We have uncovered how single-cell RNA sequencing provides detailed view of gene expression at individual cell level. I will leave the links in the description of the video that helped us to do this analysis. Thanks for watching. See you next time.